Hello and welcome to another edition of Zog Science. Today we're going to be talking about DNA. We're going to talk about kind of the discovery of DNA, what was the process, what is the structure of DNA, and then what happens when DNA replicates or when we create a copy of DNA. So just kind of start off, DNA, the discovery did not just happen at one time. If you think back to the beginning of the year, we were talking about how science is a collaborative effort. It's usually several pieces kind of put together that lead to an end result. All right? And DNA, the discovery of DNA was the exact same thing. So there was this guy named Griffith, all right, and what he was doing is he was studying the effects of pneumonia bacteria. So he had two strains of the bacteria. One of them caused lung disease and one of them did not. So what he did was that he took injected some mice with disease-causing bacteria. Obviously, they died. All right, that's what we expected. Right, that would kind of be our control, so that we could see what exactly was going on. Right, he injected some mice with the harmless bacteria. Those mice lived again as we would expect. So that would be another type of control. And then what he did was that he heat-killed some of the bacteria. So basically, he heated them up to kill them, and the mice lived. All right, so that makes sense, right? The bacteria are dead, so now they can't actually do their thing. What he then did was he mixed some of the live harmless bacteria, so these ones right here, with the heat-killed bacteria. All right, and the question is, what happened to the mice? Well, the mice died. All right, so the mice that were injected with a combination, they died as if they had been injected with the living virulent virus, all right? And when they recovered some tissue samples and analyzed it, they actually found this living virus, even though that was not or the living bacteria, even though that wasn't necessarily what they put in. So what this kind of led to is that Griffith hypothesized that basically there was something that was being passed between the dead bacteria that could kill the that were disease-causing, and they were sending something over into the harmless ones, making them deadly. All right, and we call this process transformation. So it's a process in which one strain of bacteria is changed by the genes of another bacteria. And we're going to do a transformation where we're actually going to make bacteria glow in the dark. All right, so we will explore this process of transformation more sort of in the future, and you're actually going to chance to get hands-on with that. So Avery comes along and he says, all right, I like your experiment, Griffith, but I want to change some things. So what he did was that he used enzymes to selectively destroy molecules one at a time. So he takes the lethal virus, all right, the lethal bacteria, the one that can hurt people, kills it, all right, and then he basically kind of breaks it up into various parts, the lipids, polysaccharides, right, which are also carbohydrates, proteins, and nucleic acids. He adds them to harmless bacteria, and the only one that becomes lethal is the one where the nucleic acids are left behind. All right, so those nucleic acids were known to be DNA. All right, and so what they basically discovered is that, okay, we know that DNA is what stores and transmits the genetic information. All right, so that's where they kind of got sort of the function of DNA, but now they kind of want to know, okay, what does it look like? What's the structure? So we got Hershey and Chase, all right? Again, they are going to do some experiments with bacteriophages, all right? They're going to basically kind of settle the debate whether or not it's the proteins or the DNA that carry it, because there's still at this point, even though Avery has done his experiment, there's still some question, all right? So they're just going to help provide some further evidence. What they're going to do is they're going to use radioactive elements to mark the DNA or the protein. So what they do is they basically make either the protein capsid that's on the bacteriophage or the DNA that's on the inside radioactive. So that way they can kind of track where those things go. And what they see, all right, is that only the radioactively labeled DNA is found in the bacteria cells. So that's partly how we kind of know how bacteria work, or how viruses work. You know, we talked about that a little bit in our last unit, right? But they found that the DNA is inside, right, inside the cells, but it's not in the supernatant. So basically they figured out it's inside the cells, not somewhere else. They basically kind of supported the conclusions of Avery and specified that the genetic material is DNA, and that's what's being passed on from one generation to the next. Then we've got this guy named Chargaff. All right, so again, at this point we have determined that DNA is the genetic information. All right, and Chargaff's going to come along and he's going to do some experiments that are going to help with the structure. So he determines that in any sample of DNA, so if we take any random sample of DNA, whether it be from humans, bacteria, plants, whatever, that the number of adenines is equal to the number of thymines, and that the number of cytosines is equal to the number of guanines. All right? So what is kind of the logical conclusion from this? Well, they conclude that A and T must be paired together, because if for every one adenine there's one thymine, and for every one cytosine there's one guanine, so G and C must pair together as well. We've also got a lady at the time named Rosalind Franklin, and she uses X-ray diffraction to create pictures of DNA's molecular structure. All right, and that's the kind of the famous picture that she comes up with. Unfortunately, due to her work with X-ray diffraction, does involve a lot of radiation, and she dies only a few years later from cancer. And she never, at the time, didn't really get recognized for her efforts. Later on, as kind of time has passed, she's been recognized for the efforts that she put in, but at the time she kind of got shafted a little bit, and that's because there's these two guys named Watson and Crick. So Watson and Crick determined the structure of DNA in 1953, and they used their work and the work of the previous scientists, and they also kind of took a sneak peek at Franklin's X-ray images before they were published for the public to be able to see for all the rest of the scientists. So they kind of got a head start. And they didn't really ever give Rosalind the kind of credit that she really deserved until much later on. So the DNA structure was determined to be shaped like a double helix. And if you look at that X-ray photo, right, you can kind of see here why you would think that, right? Because you've got this kind of X right here, right? And that's where you've kind of got the spinning occurring of that double helix. And again, you've got bonds, hydrogen bonds forming between the A's, T's, G's, and C's, and we're going to take a look at what those hydrogen bonds are later on. So what we're going to do right now is you're going to draw a diagram of, all right, so here's again that double helix structure, that spinning. Here's where you can kind of see where that X would be. So I'm going to go ahead and put this up and leave it for you. All right, so I want you to draw this picture. Go ahead and pause the video and draw the picture. All right, moving forward. All right, DNA replication, okay? So DNA has to be replicated or copied in order to ensure that during the cell cycle, each daughter cell receives a complete copy, all right? So when we go through mitosis, which we just discussed, right, we've got two daughter cells that are going to be genetically identical to one another. And in order for that to occur, we have to have our DNA replicated, all right, or essentially copied. All right, so how does that process occur? Well, remember, it occurs during the S phase of the cell cycle, right? Before the chromatin, remember, which is the DNA that's kind of spread out in the cell, condenses into the chromosomes. All right, and this kind of shows you how DNA looks when it's kind of spread out. There's these proteins that it kind of wraps around, and then it coils up and forms a chromosome, and the centromere is kind of in the middle. So DNA replication occurs in 4 basic steps. Step 1, all right, we've got this protein enzyme, okay, notice the ACE ending. We got an enzyme, all right, that comes along, and it's going to break open, essentially unzip the hydrogen bonds between the base pairs. This is part of the reason why the hydrogen bonds are so important in DNA. It helps to hold it together, but because they're only hydrogen bonds, we can easily break them apart in order to do replication. Step 2, we've got DNA polymerase 3. Again, this is an enzyme, all right? It's going to insert the appropriate bases. So we've got our template strand, right, the original DNA, and if there's an A on the template strand, then the new strand is going to add a T. And if we have a G, then it will add a C, all right? So DNA polymerase kind of comes along, and it's going to be adding those on. Now, one of the things you kind of notice is that there's 2 different directions here, okay? So DNA has a direction that it goes in. The strands kind of go opposite to one another. So one side, we call this the leading strand. Okay, it is replicated consistently all the time. And then the other strand down here, we call this the lagging strand. So you notice that there's kind of 2 polymerase. There's kind of these 2 like pieces right here. All right. So the leading strand can go continuously, but the lagging strand, because of the directionality here, it can only go in this direction And so it's only able to add on at a short amount of time, add on some at a time as the helicase unzips it. All right. So yep, DNA polymerase 3 adds stuff on. Step 3, a new sugar phosphate backbone is built. So we got our new sugar phosphate backbone being built here and here. And then 4, the sequence is going to be proofread by DNA polymerase 1. And DNA polymerase 1 is also going to connect these fragments right here. We call these Okazaki fragments, O-K-A-Z-I, O-K-A-Z-A-K-I, sorry, O-K-A-Z-A-K-I, Okazaki fragments. And those are kind of the pieces that are created by the DNA polymerase 3. DNA polymerase 1 comes in proofreads. And the reason why we proofread is so that we don't get mutations that occur. Mutations can lead to cancer. So DNA polymerase 1 is very important in its job that it does. DNA replication is considered to be semi-conservative because each side of the parent strand serves as a template for the new strand. So each basically new molecule of DNA is composed of half old and half new DNA. So that's why we call it semi-conservative. Some of it is conserved, but not all of it. So let's do some base pairing practice. All right, so I'm just gonna give you 10 seconds or so to think about what's gonna come up next. So if I've got TTGCTAG, what should it come up with? All right, and the correct answer would be AACGATC. All right, if we've got this TAGCGCT, we would have ATCGCGA, and the last one, ACCGTCA. We would have TGGCAGT. All right, well, I believe that that's about, oh, we got one more, sorry. GCTATGT. And there's your answer. So it's fairly simple. A's go with T's, G's go with C's. All right, everybody, I hope you have a good day, and thanks for watching.